Hello. Welcome to next in the series review clips designed to assist you in preparation for your Biology 301 public examination. In this clip, we'll be taking a more detailed look at diagnosis and treatment of genetic disorders. There are a number of different means by which genetic disorders can be detected. These include, firstly, amniocentesis. With amniocentesis, fluid is drawn from the amniotic sac with a needle. Following the drawing of the fluid, a karyotype is prepared along with other genetic tests to determine disorders. This can be done because with the onset of amniocentesis, if you take this oval to be the woman's abdomen, inside of the woman's abdomen is a sac which contains the fetus. A long needle will be drawn into the amniotic sac and fluid withdrawn from it. This fluid will contain some fetal cells. These fetal cells can be analyzed for chromosomal abnormalities through a karyotype. Another means by which genetic disorders can be determined, is through the use of a chorionic villus sampling. Here, cells are removed from the chorion or placenta, and in a similar fashion, a karyotype can be deducted. Again, this will indicate genetic disorders. With regards to a chorion villus sampling, if we take this red oval to be the abdomen, inside of the abdomen, inside of the amnion, is the amniotic sac, the fetus is contained within the amniotic sac, of course, and from that there are a number of layers, one of which is the chorion, which I'll draw here in red. And of course the yolk sac as well, in blue. Through the use of a needle, this here comprised together forms a placenta. Cells are drawn from the placenta using a needle, which can be inserted either through the abdomen or intravaginally. In both cases, cells can be drawn for further analysis. These fetal cells, these cells from the chorion, give an indication of the genetic makeup of the fetus as well. Another tool is through the use of fetoscopy. Fetoscopy and fetoscopy is exactly that. A camera is inserted, typically vaginally, to view the fetus, to look for a physical abnormality that may suggest genetic disorders. Genetic markers. Once the DNA is determined and is drawn from either amniocentesis or CVS, this can indicate the inheritance of a defective gene or a gene causing disorders. There are two types of these markers. The first of these are link markers, and link markers are a known sequence of nucleotides located close to a gene that causes a disorder. The other type are the gene-specific markers, and these are a sequence of DNA that is part of the gene itself. Both of these, used in concert with each other, are found using radioactive fluorescent tags on a complementary DNA nucleotide sequence of the suspected marker. Once disorders are detected, there are several methods of treatment that are available. One of these is genetic screening and prevention, and here blood and other biochemical tests are done either at birth or prior to determine the disorder. Once the disorder is detected through screening and prevention, parents would be advised of available options such as diets in the case of PKU that they could place their children on. Surgery is an option for genetic disorders which cause physical abnormalities. Cleft palate will be one example. A surgical procedure to repair the cleft palate would help alleviate some of the concerns with this genetic disorder. Environmental control can be done as well. This simply is controlling the environment which an individual must cope with. Albinism. They must avoid sunlight. Usually they're fully clothed all the time and only good at night, and those sorts of things. And certainly gene therapy may be an option. Here, normal or modified genes are transferred into the defective cells in an individual. Ideally, these new genes should cause the cell to work normally and fix the genetic disorder that is present. Now, with gene therapy, there undoubtedly are many questions that arise from its use. Are we playing God? Who gets to modify a gene? Who does not? Under what conditions are genes produced and used? What costs and who pays for the procedure? All of these are ethical questions that need to be answered and need to be considered. Past public examination have asked you to offer your opinions on these very types of questions. By way of a sample for questions that appear from this material, I present the following. A 42-year-old woman who is 8 weeks pregnant is concerned about the possibility of genetic abnormalities in the fetus and wants to be tested immediately. Which is the best technique to use? Because you're interested in the genetic abnormalities of the fetus, you need to get a genetic sample. Photoscopy and ultrasound would not be able to provide that. As this is at 8 weeks, using an amniocentesis, inserting a needle, would be quite risky. For this reason, the correct choice is B, chorionic villi sampling. This second question, again for material on past public examinations. Which two techniques are used to diagnose prenatal chromosomal abnormalities? Again, prenatal chromosomal abnormalities. Photoscopy and ultrasound. These could be used to get pictures, but they cannot definitively diagnose chromosomal abnormalities. Genetic counseling and genetic markers, they'll be done post-birth. But again, genetic counseling is certainly after birth with the parent. Karyotyping and gel electrophoresis does give an idea of what's there, but the correct answer turn technique used, this would be A, amniocentesis and chorionic villus sampling. And this question is posed, ask which technique enables direct observation of a developing fetus? Genetic markers, amniocentesis, and chorionic villus sampling all involve looking at the genetics. It is through photoscopy that doctors can get an exact picture of the fetus. Thank you very much for your patience. I hope you found this learning clip to be helpful.